Imagine a serial killer that has been, hunting humanity for millennia.
Right.
A killer so pervasive and completely invisible that for 2,000 years, the smartest doctors on Earth just blamed the air itself.
Literally just the air.
Exactly. So today, we are bringing you a deep dive into Plasmodium. Okay, let's unpack this because we aren't just talking about a bug here. We're talking about the architect of a disease that has shaped human history, altered our DNA, and, it remains an overwhelming global force today.
Yeah, it is arguably one of the most consequential organisms on the planet. And when we say Plasmodium, we're referring to a genus of single-celled parasites that cause malaria.
Right.
To really put the stakes of this deep dive into perspective for y'all, this single genus is responsible for roughly 500,000 deaths and 500 million clinical cases every single year.
It's just staggering. 500 million.
And the most sobering aspect of that statistic is who actually pays the price. The mortality burden overwhelmingly falls on children under 5 in sub-Saharan Africa.
Wow.
Yeah, the sheer scale of it is just immense.
The numbers are, frankly, difficult to fully comprehend. But diving into our sources, the historical blind spot surrounding this disease is fascinating.
Oh, absolutely.
Because for millennia, humanity was completely wrong about what was actually causing this massive death toll. The word malaria literally translates from Italian as bad air, malaria.
Yeah.
People firmly believed the sickness was caused by these toxic miasmas, which were essentially, noxious vapors rising out of swampy areas.
It's a classic example of correlation being mistaken for causation, right?
Definitely.
The ancient observers, they weren't entirely wrong to suspect the swamps. The geography was highly relevant, but not because of the air. It was because the stagnant water provided the perfect breeding ground for the true vector of the disease.
But to actually see the killer, to understand how it works, we had to transition away from ancient Roman theories and essentially look through a 19th century microscope.
Exactly.
So picture French colonial Algeria in 1880. There's this French army physician named Charles-Louis Alphonse Leveron, and he's working in a military hospital.
Right, looking at blood samples.
Yeah, he's examining the blood of a soldier suffering from these intense cyclical fevers. And at the time, other doctors were seeing this strange black pigment in the blood of fever patients and just writing it off as degeneration.
Like the blood was just breaking down.
Exactly. That was the consensus. But Leveron looks closer, and he notices these crescent-shaped bodies within the blood, and more importantly, they have moving flagella. They're actively swimming.
Wow.
He realizes, hey, this isn't decaying blood, it's a living parasite.
And that observation fundamentally shattered a 2,000-year-old paradigm. He rightly won a Nobel Prize for it in 1907, but Leveron only solved half the puzzle, right?
Right. Finding it doesn't explain how it got there.
Exactly. Finding the parasite in human blood doesn't explain the transmission. For that piece, we have to fast forward to 1897 in India. There's a British medical officer named Ronald Ross. He's building on a hypothesis from his mentor, and he takes a female Anopheles mosquito that had fed on a malaria patient, and he dissects its stomach. And there it is. The parasite is growing right there in the mosquito's gut wall.
And the historical drama behind that discovery is remarkably intense in the sources.
Oh, it's messy.
Very messy. You've got this bitter priority dispute between the British school, led by Ross, and the Italian school, led by Giovanni Battista Grassi.
Right.
Grassi and his team were doing this incredibly meticulous work, demonstrating the complete mosquito-to-human transmission cycle for human malaria, using Anopheles claviger mosquitoes.
Which is key.
But Ross, on the other hand, was using bird malaria in sparrows, because, experimenting on humans was fraught with ethical complications.
Sure.
Yet ends up winning the Nobel Prize in 1902 and effectively secures the public narrative, even though Grassi's evidence was far more applicable to human disease.
Yeah, that rivalry really set a template for the field. Credit for major Plasmodium discoveries has repeatedly been contested throughout history.
Hold on, though. Before we go further down the history rabbit hole, I need to ask a structural question about the sources.
Sure.
What actually is this parasite? Because when we hear single-celled, we usually default to bacteria. Or we hear infectious disease and we think, oh, a virus. But Plasmodium is neither of those things, right?
What's fascinating here is just how evolutionarily advanced this organism is compared to a bacteria or a virus.
Really?
Yeah. Plasmodium is a eukaryotic protist. It belongs to a phylum called Apicomplexa. Because it's a eukaryote, it is actually much closer to us, biologically speaking, than it is to a bacterium.
That is wild.
I know. It has a defined nucleus, it has mitochondria.
And it has that bizarre feature mentioned in the genetic research, the apicoplast.
Yes.
If I'm understanding the secondary endosymbiosis concept correctly from the sources,
Mhm.
this means that millions of years ago, the ancestor of this parasite essentially swallowed a red alga, like a plant-like cell.
Right.
And instead of digesting it, it just incorporated it into its own biology.
That is an excellent summary.
Yeah.
The epicoplast is a vestigial, non-photosynthetic organelle inside the parasite.
Wow.
It provides hard genetic evidence of that ancient evolutionary merger, so it can't perform photosynthesis anymore, obviously, but the parasite still relies on the metabolic functions of this captured plant part just to survive.
So we are dealing with a shape-shifting, single-celled organism operating with ancient plant parts inside of it.
Pretty much.
That level of complexity explains why it doesn't just enter the body and immediately start copying itself like a simple virus does.
No, not at all.
It goes on this highly orchestrated, multi-stage journey through different organs. So let's trace the life cycle, because basically every symptom and every medical intervention maps directly onto this journey.
Okay. Stage 1 is the initial transmission.
The bite.
The bite, exactly. When a female Anopheles mosquito takes a blood meal, she injects saliva into the skin to prevent the blood from clotting.
Gross, but effective.
Right. And hidden within that saliva are the infectious forms of the parasite called sporozoites. They are thread-like and incredibly mobile, and we're talking about a microscopic invasion force here. Often just a few dozen sporozoites are injected during a single bite.
A few dozen. That's it.
That's it. And within roughly 30 minutes, they have completely vanished from the bloodstream.
Wait, if they vanish from the blood in 30 minutes, the body's immune system doesn't even have time to mount a proper response. Where are they hiding?
They travel directly to the liver. This initiates stage 2, the liver stage, which in a literature is referred to as exoerythrocytic schizogony.
Oh, exoerythrocytic schizogony. Medical terminology really knows how to clear a room.
It's a mouthful.
Let's just call it the liver invasion.
Yeah.
But the stealth aspect of this invasion is incredible. The sporozoids that glide through cells, find a capillary, and specifically seek out hepatocytes in the liver.
Right.
Once inside the liver cell, they multiply asexually. One sporozoid can produce tens of thousands of daughter cells, which are now called merozoites.
Tens of thousands.
And while this massive army is quietly massing inside the liver for 1 to 2 weeks, the patient feels absolutely nothing. Zero symptoms.
The parasite is entirely bypassing the immune system's early warning radar. It's like a stealth bomber.
Yeah.
And this brings us to a terrifying quirk associated with 2 specific species, Plasmodium vivax and Plasmodium ovale.
Okay, what do?
In these variations, some of the parasites in the liver don't divide immediately. They arrest their development and turn into what are called hypnozoites, basically dormant sleeper cells.
So they just sit there, completely undetected.
For months, sometimes even years.
Are you kidding now?
This means a person could travel to an endemic area, return home, test negative, feel perfectly fine for a year, and then suddenly develop full-blown malaria without ever being bitten by another mosquito.
That is deeply insidious.
The parasite simply reactivates its life cycle on a delayed timer.
Okay, but eventually, whether it's an immediate cycle or a delayed reactivation, the army moves out. And that brings us to stage 3, the blood stage. The merozoites burst out of the liver and actively invade red blood cells.
This is where things get bad.
Yeah, once inside the red blood cell, they consume the host hemoglobin, multiply another 6 to 20 times, and then blow the cell apart to release the next generation to invade fresh blood cells.
And this stage, this blood stage, is what actually causes the clinical illness.
Right.
Because when those red blood cells rupture, it isn't just releasing new parasites, it is dumping parasite waste products, toxins, and cellular debris straight into the bloodstream.
Wow.
This triggers a massive, explosive, inflammatory immune response. And because the parasites synchronize their multiplication, a huge portion of infected cells rupture at roughly the exact same time.
So that synchronized cellular explosion is what causes the hallmark symptom of malaria, the intense cyclical fevers.
Exactly.
Here's where it gets really interesting, because the timing of those fevers historically acted like a diagnostic fingerprint.
Oh, for sure.
Plasmodium vivax, ovale, and the deadly falciparum cycle roughly every 48 hours. Plasmodium malariae cycles every 72 hours. But the danger isn't just the bursting of the cell, right? It's what falciparum specifically does right before it bursts.
Yes, this is a crucial mechanism of disease severity. Plasmodium falciparum possesses a family of genes called the VAR genes.
Okay.
These genes encode for a specialized surface protein known as PFEMP1. The parasite transports this protein to the surface of the infected human red blood cell, causing it to form these little physical knobs.
And those knobs essentially turn the infected red blood cell into microscopic Velcro.
That is the perfect analogy. It makes the infected red blood cells incredibly sticky, a process called cytoadherence. The blood cells latch onto the endothelial linings of your blood vessels.
The underlying logic of that is brilliant from a survival standpoint,
It really is.
because the spleen acts as the body's blood filter, constantly surveying for damaged or infected red blood cells. If the infected cell floats freely through the bloodstream, it will eventually pass through the spleen and be destroyed.
Right.
But by sticking to the blood vessel walls, the parasite physically anchors itself outside of the circulation, entirely hiding from the spleen. But the consequence for the human is devastating.
Devastating.
Yeah.
Because these sticky cells clump together and block blood flow in the microvasculature.
Oh, no.
When this clogging happens in the delicate vessels of the brain, it causes cerebral malaria, which results in swelling, coma, and is rapidly fatal if not treated immediately.
It's a masterclass in biological engineering.
Truly.
And finally, stage 4. A small percentage of these blood-stage parasites don't replicate asexually. They transform into male and female forms called gametocytes.
Right, the reproductive stage.
They stop dividing and just float in the blood, waiting. Because their sole biological purpose is to get sucked up by the next mosquito that happens to bite the host.
Yep.
Once inside the mosquito's midgut, they mate, burrow into the gut wall, multiply, and eventually travel to the mosquito's salivary glands to start the whole cycle over again.
It is a relentless mechanism, and it has been repeating with such lethal pressure over thousands of years that it has literally forced human DNA to change in order to survive it.
Wow.
Which brings us to the evolutionary arms race between humans and Plasmodium.
Yeah, if this brutal cycle has been killing people for thousands of years, our biology had to find a way to fight back.
Absolutely.
Let's talk about Anthony Allison's 1954 study in Kenya. He was investigating the sickle cell gene. Now, usually if someone inherits 2 copies of this gene, they develop sickle cell disease, which alters the shape of red blood cells into crescents, leading to severe, painful, and life-threatening complications.
Right.
But Allison discovered that if a person only inherits 1 copy of the gene, a condition called sickle cell trait, they suffer dramatically less severe falciparum malaria.
The physiological mechanism is incredible. When the parasite invades a red blood cell in someone with sickle cell trait, the cell tends to sickle and collapse under the stress.
It just caves in on itself.
Exactly. This structural collapse, along with the low oxygen environment it creates inside the cell, physically impairs the parasite's metabolism and its ability to thrive and multiply.
That's amazing.
It is the textbook example of what evolutionary biologists term balancing selection. A gene that is highly detrimental in a double dose is maintained at a high frequency within a population because inheriting a single dose offers life-saving protection against a much deadlier environmental threat, which in this case is endemic malaria.
But there's another genetic defense detailed in the sources that I find even more fascinating: Duffy negativity.
Oh, yes.
So Plasmodium vivax needs to bind to a very specific receptor to break into a human red blood cell. It's called the Duffy antigen.
Right.
But the vast majority of people of West and Central African descent simply do not express this antigen on their red blood cells.
It functions as a localized evolutionary shield.
It's like the parasite spent millions of years forging the perfect skeleton key to rob a bank, and human evolution responded by just changing the locks on the vault.
I love that analogy.
The key just no longer works. An entire species of parasite is physically locked out of the blood cells of an entire human population. That is the main reason vivax is so rare in sub-Saharan Africa today.
But the parasite is highly adaptable. It's not just human genetics that are evolving. The boundaries of the disease itself are shifting.
Okay, how so?
For decades, the scientific community thought of malaria as a fixed set of human-specific pathogens. But in the early 2000s, a Malaysian researcher named Balbir Singh was investigating what appeared to be a cluster of Plasmodium malaria infections in Sarawak, Borneo.
But his molecular analysis showed it was something else entirely, right?
Precisely. Through genetic sequencing, his team proved it was actually Plasmodium knowlesi.
Okay.
This was considered a parasite that exclusively infected monkeys, but it had made the zoonotic jump to humans.
Well,
And unlike other human malarias, Nalesi cycles in the blood every 24 hours, meaning the parasitemia can spike and the disease can progress incredibly fast.
and the sources point out that this zoonotic jump is largely driven by deforestation.
Yeah, unfortunately.
As forests are cleared for agriculture or development, it brings human populations, macaques, and the mosquitoes that feed on both of them into much closer proximity. We are essentially building the ecological bridge for the parasite to cross over into our species.
The environmental interactions change, and a monkey parasite suddenly becomes a dangerous new threat to public health.
Which perfectly illustrates why you can't just wait for evolution to solve the problem. You have to use chemistry.
Right.
Since the 20th century, humanity has actively tried to eradicate this parasite using science and medicine. And it has led to some of our most brilliant scientific triumphs and some spectacular failures. Let's look at the story of 2UU and Project 523.
A truly remarkable piece of medical history. The year is 1972, against the backdrop of the Vietnam War. North Vietnamese soldiers were dying of malaria at alarming rates because the falciparum parasite was evolving resistance to chloroquine, which was the standard anti-malarial drug at the time.
Right. In response, the Chinese military launches this highly secretive initiative called Project 523 to find a new cure.
And 2UU is a pharmacologist assigned to the project. She doesn't have a PhD, nor does she have medical training outside of China.
But what she does have is an incredible dedication to screening traditional Chinese medicine texts.
Exactly.
After screening thousands of recipes, she zeroes in on sweet wormwood, Artemisia annua, but her initial chemical extracts of the plant are inconsistent. They aren't reliably curing the malaria models in the lab.
And this is where her deep dive into historical texts paid off. She found a 4th century manual of clinical practice by an alchemist named Ji Hong.
Yep.
The text specifically instructed to soak the wormwood in water and wring it out to obtain the juice. It was a cold extraction method.
Right, because standard laboratory extraction involved boiling the plant material. You realize that the heat from the boiling process was actually breaking the delicate molecular bonds of the active compound.
It was completely destroying its medicinal properties before they could even test it.
Exactly.
Yeah.
By switching to a cold extraction method using ether, she successfully isolated artemisinin. This compound works by reacting with the iron inside the parasite, creating free radicals that destroy the parasite from the inside out.
It's brilliant.
Artemisinin became the absolute backbone of global malaria treatment. Decades later, in 2015, she won the Nobel Prize in Medicine for a discovery rooted in ancient texts.
It is a phenomenal story. But unfortunately, the evolutionary arms race continues. We are now seeing the spread of artemisinin resistance, particularly in the greater Mekong region of Southeast Asia.
Oh, well, that's not good.
Parasites carrying specific mutations, notably in the Kelch13 gene, are surviving the initial drug onslaught, leading to delayed clearance times in patients.
Wow.
This is widely considered one of the most alarming developments in modern infectious disease because right now we do not have a fully proven successor drug ready to replace artemisinin-based combination therapies on a global scale.
We are constantly one step behind. And we've seen what happens when we underestimate the complexity of this disease.
Oh, definitely.
Look at the Global Malaria Eradication Program, launched by the World Health Organization between 1955 and 1969. The strategy was heavy reliance on indoor residual spraying of the insecticide DDT to kill the mosquitoes before they could transmit the parasite.
And, well, to be fair to the historical context, that strategy was highly successful in certain environments. It helped eliminate endemic malaria from Europe, the United States, and parts of the Caribbean.
But why did they assume a one-size-fits-all approach would work globally? I mean, an intervention built for suburban America or temperate Europe is obviously going to face entirely different logistical and biological challenges in a dense tropical ecosystem. It seems incredibly naive in hindsight.
The ecological mismatch was profound. The program failed catastrophically in sub-Saharan Africa.
Right.
The higher density of mosquito populations, the much longer year-round transmission seasons, and the lack of robust health infrastructure made it an unworkable approach for that specific environment.
And the mosquitoes adapted, right?
Yeah. Worse, yeah. The mosquitoes began evolving physiological and behavioral resistance to the DDT. When the eradication program was officially abandoned in 1969, the resulting pessimism severely stifled international funding for malaria control for decades.
It's a harsh lesson in biological humility. And we're seeing similar tensions today when we look at the cutting-edge tools currently in development, specifically vaccines and gene drives.
Yes.
The sources present some very intense, complex debates around these technologies, and it's important we lay out the core of these arguments impartially for you. Let's start with vaccines. Why did it take until 2021 to get the first WHO-recommended malaria vaccine, RTSS?
It fundamentally goes back to what we discussed earlier about Plasmodium being a complex eukaryotic protist. It has a multi-stage life cycle, shifting its form from the skin to the liver to the blood.
It's a shapeshifter.
Exactly. And each stage, it expresses different proteins, essentially changing its disguise to hide from the immune system. Developing a vaccine against a shape-shifting organism is exponentially more difficult than targeting a relatively stable, simple virus.
Makes sense.
As a result, the new vaccines, RTSS and the newer R21, only offer partial efficacy, ranging somewhere between 30 to 75%, depending on the dosing schedule and the specific trial conditions.
If you're listening to this and thinking, why bother with a vaccine that only works 30 to 75% of the time, you aren't alone. That exact question tore the global health community apart.
Yeah, it was a massive debate.
On one side, advocates argue that even 30% efficacy against a disease that kills hundreds of thousands of children every year is a massive victory in absolute numbers and is absolutely worth deploying.
Right.
But on the other side, critics question the resource allocation. They point out the massive financial cost and the logistical nightmare of delivering a multi-dose vaccine in remote rural areas. They argue those funds might save more lives if spent on cheap, highly proven interventions like insecticide-treated bed nets, which are currently underfunded.
If we connect this to the bigger picture, it highlights just how difficult public health decisions are when resources are finite.
Exactly. But the debate around CRISPR gene drives is even more profound.
Oh, absolutely. Research consortiums like Target Malaria are utilizing CRISPR technology to engineer gene drives in Anopheles gambia mosquitoes.
How does that work?
Well, a normal genetic trait has a 50% chance of being passed to offspring. A gene drive overrides traditional genetics, forcing a specific trait, like female sterility, to be inherited by nearly 100% of the offspring.
Wow.
The goal is to release these genetically modified mosquitoes into the wild, where they will mate and pass on this sterility trait, eventually crashing the wild mosquito population entirely.
They are essentially engineering extinction.
Yes.
And the divide on this is massive in the sources. Proponents argue that we have a moral imperative to use every tool at our disposal to eradicate a diseased vector responsible for unimaginable human suffering. The potential to permanently save millions of lives is sitting right there in a lab.
But the critics raise massive red flags regarding the ecological consequences. They argue that intentionally releasing a self-propagating genetic change into a wild ecosystem is irreversible. If we intentionally wipe out a foundational species like a mosquito, we simply don't know the cascading impact on the food web.
What happens to the fish, birds, and insects that rely on them? We are impartially presenting both sides here, but it is undoubtedly one of the most consequential scientific debates of our lifetime.
It requires society to decide what level of ecological uncertainty is acceptable when millions of human lives are on the line.
So what does this all mean? We've journeyed from ancient theories of swamp miasmas to the bleeding edge of CRISPR gene drives. But why is it so critical that you, listening to this right now, understand the biology of Plasmodium today?
Because malaria is not a static disease of the past. It remains a glaring issue of global health equity.
Yeah.
We have a disease that is largely preventable and treatable, yet it continues to kill hundreds of thousands of people simply because they reside in the world's poorest regions.
It's tragic.
It is. Furthermore, the map of the disease is changing. As climate change alters global temperatures, the geographic range where Anopheles mosquitoes can survive and transmit the parasite is expanding.
Right.
We are already seeing malaria transmission moving into higher altitude regions in East Africa, places that historically used to be too cool for the parasite to thrive. The territory of this disease is actively growing.
It is a highly dynamic, evolving threat. And there's exactly why we do these deep dives.
Yep.
Before we go, I want to leave you with one final provocative thought based on the research we unpacked today.
Okay.
Remember that apicoplast we talked about in the very beginning? That vestigial plant-like organelle the parasite absorbed from red algae millions of years ago?
The ancient evolutionary relic.
Exactly. Well, human biology is strictly animal. Our cells do not have anything even close to a plant organelle. So if pharmaceutical researchers can design a drug that specifically targets the metabolic machinery of that
Mm.
ancient alien piece of plant life hiding inside the parasite, could that be the ultimate Achilles heel?
That's a fascinating angle.
Right. Could we find a way to completely destroy Plasmodium without harming a single human cell in the process? It's something to think about. Thank you so much for joining us on this deep dive. Keep questioning the world around you, and we'll see you next time.